So let's pick up where I left off last time, which was with a really neat experiment called the Fry and Edidin experiment. You don't need to know the names, just the names of the two investigators at Johns Hopkins University who did this experiment about 40 years ago. Just to repeat the setup for the experiment, they took two cultured skin cells grown in the laboratory in Petri plates. One was human cells, the other was mouse cells. They produced antibodies against human proteins that were linked to a dye. Not antibodies made in a human, antibodies made in a rabbit against human cells. And they made antibodies against the mouse surface antigens labeled with a different dye called rhodamine that fluoresces red. And then the object was to cause the two cells to fuse so that the plasma membranes would surround the cytoplasms of both cells and the nuclei would come in together to make a heterokaryon. And then, at times, after the fusion of these cells, they asked, where are the green antigens and where are the red antigens? Do they stay in their separate halves or do they mingle? And the rate at which they mingle, is that a reflection of some state of the phospholipids in the membrane? So how do you bring these cells together? Well, one technique that's widely used is the use of a virus that itself has a membrane. It's called Sendai virus. And it's got an RNA genome, and it's kind of a vesicle surrounding it with membrane proteins that allow the virus to fuse at the cell surface of a cell it's about to inject and inject the RNA core. But if you have a high concentration of cells that are more or less touching each other and you add the virus, then occasionally the virus will cause the two cells to fuse with one another, making a junction across which membrane proteins and lipids may flow. So initially this fused cell looks like a kind of a dumbbell-shaped structure, but if the heterokaryon is incubated at physiologic temperature for a few minutes, it rounds up and it's much bigger. And then with time, you apply these yellow, green, and red linked antibodies, and you ask where are the antigens to which the antibodies bind. Initially, when the samples are first fused, the green antigens are on one side and the red on the other. But with time and incubation at physiologic temperature, the signals merge, and the rate at which they merge is roughly predicted by the viscosity of the phospholipids that you extract from the membranes from these cells. If you cool the sample after fusion on ice, then the antigens don't mix. If you add an energy poison to block the motile apparatus within the cell, the cytoskeleton, and warm the sample up, they still mix. So you don't require any ATP-driven active processes to cause the merging of these two populations of antigens. All you need is that the lipid matrix be at or above its phase transition temperature, its T sub m. So this experiment then, in a rather crude way, illustrated the mobility of proteins within membranes. It's crude because the antibodies recognize dozens of different protein and carbohydrate molecules, so one can only compute a kind of an average diffusion of a population of proteins. So it's not terribly precise. Of course, from what I told you last time, it's also possible to do this experiment with an antibody against an individual pure plasma membrane protein. If you have a plasma membrane protein that you've isolated and raised an antibody against that, you can repeat the same experiment, and then the measurements become somewhat more refined because you're now measuring the behavior of an individual rather than a collection of molecules. Okay, so but that's still a relatively crude measurement. It requires that you manipulate these cells and get them to fuse, and it's a little messy. Maybe the fusion with a virus induces some kind of mobility in these membranes that's unnatural. Well, now let me tell you about another technique, a rather, again, a really more sophisticated technique, one that's only really become possible, let's say, in the last 10 or 15 years. And that relies on a fluorescent antibody, just as I've said before, that binds to a particular protein on the surface of a cell. And then it further relies on the ability to bleach a patch of membrane with a laser pinpoint right on the surface of the cell. So you can focus a laser beam with enough energy to bleach a restricted region of fluorescent molecules without frying the cell. You can actually do this on living cells. So you shine a pinpoint of laser light at the excitation maximum for a particular fluorescent probe, but with such intensity that the probe bleaches rather than just fluoresces. So it bleaches. And those molecules then go dark. You don't see them. And so as a consequence, you see a kind of a dark spot in a sea of a fluorescent haze on the rest of the surface of the cell. Now, since the laser is tuned at an intensity that doesn't fry the membrane, it doesn't poke a hole in the membrane containing these bleached fluorescent probes is still alive, and fluorescent molecules from the neighborhood can diffuse in, and bleached antibodies and their complexes can diffuse out, so that with time, and a rate that can be measured very precisely, looking right at that blank spot, with time, the signal merges so that now the blank spot disappears. This technique is called fluorescence recovery after photobleaching, or FRAP for short. It's a very important technique because it can be tuned to tiny areas on cells, or even within cells. You can shine a light on an intracellular organelle, bleach all of the fluorescent, for instance, GFP molecules in a particular organelle, and watch as new GFP molecules move into that organelle. So it's really a wonderful technique, and it can be applied to living cells. So this then allows one a very precise measure of the rate of diffusion. And again, the calculations show that for most molecules on the cell surface, they're free to move about, limited only by the viscosity of the matrix of the membrane. Yes?
When you're trying to calculate the rate of diffusion
As the rate of the local temperature
of the cell,
Yes.
they also try and normalize it by finding out how much absorption there is to the laser, how much energy is going in.
Yeah.
So I'd assume the laser would locally heat the cell.
Yeah, that's a good question. All I can tell you, all I know is that it's done under conditions that don't damage. And any heat that's generated locally would probably dissipate very quickly. And in any case, of course, if you turn the amplitude up, you can actually poke a hole in the cell too. You can kill cells this way, obviously, but it's done so as to do as little damage as possible. And the fact that you get this disappearance within a very brief period of time that is temperature dependent says that the membrane has withstood the insult of this laser attack. Okay. All right, so now let me emphasize again, this experiment works for most proteins, but there's really an interesting difference for some proteins. And so if you would turn to the handout from last time and look at the last diagram page where there's a kind of a cartoon of the a kind of cross-section looking down onto the cytoplasmic face of the red blood cell membrane. Let me tell you that there is I remind you that there's this network of spectrin. It's plastered along the inner surface of the red cell membrane. It is polymerized to create a biconcave shape. And of course, this network must be anchored at certain points on the cell surface. So there must be something to hold it in place. And that thing that it's holding it in place is a protein in the membrane called initially it was called band 3, simply because it was the third protein down on an SDS gel, but it turns out to be a channel protein that transports ions bicarbonate into cells, into and out of red cells. Anion transport channel. So it's a membrane protein that spans the bilayer many times. It has some determinants facing the cell surface, and it has other determinants facing the cytoplasmic surface, and at least one of those sites on the inner surface of the membrane, the plasma membrane of the red cell, makes direct contact with the spectrin network. And so the network is held in place by being registered at this site. So now let's do another kind of a thought experiment, actually a real experiment, but I'm just going to draw it on the board. And suppose I told you that we had an antibody against the band 3 anion transport channel, and we made that fluorescent with a red dye. And it's uniform because there are many thousands of copies of this protein in the membrane. And we used a laser tuned to the excitation frequency of this dye, and we get a dark spot. Given what I've just said, what would you expect to happen with time to that dark spot? Somebody raise their hand.
It's attached to the spectrum.
Yes.
So
nothing would happen. Be more specific. Nothing would happen.
So you wouldn't see the fluorescence return.
Good. So you would not see, or it would be much slower FRAP recovery, because the protein is anchored in place and it can't move about. You can do this experiment in a really neat way. You could do it with two different dyes. You could do it with a red dye linked to the antibody against the anion transport channel, and a yellow dye linked to the glycophorin molecule. You can bleach in two spots on the same cell, and one spot will stay dark, and the other spot will recover within a brief period of time. And so, of course, as the answer indicates, it's because that protein is held in place. And so this set of observations leads to a general model of membrane structure, which is true to a large extent, but not entirely. That was posed, again, about 40 years ago, called the fluid mosaic model of membrane structure. And this model proposes that most membranes are two-dimensional fluids, and that proteins are free to move about, limited only by the viscosity of the matrix that they encounter, and they are not organized in any way unless they are tethered by some extrinsic factor. This being, for instance, spectrin in the case of red blood cells, or the actin network, as you'll see extensively in nucleated cells. So no order, free to move about, with 2 degrees of freedom. Proteins and lipids can diffuse, translate within the plane of the membrane in 2 dimensions, or they can spin about an axis perpendicular to the plane of the membrane, but they are not free to dip back and forth, to flip-flop in a transbilayer manner, because the external polar portion or the internal polar portion of a protein cannot readily embed back into the bilayer once it's achieved its asymmetric orientation through biosynthesis, which is what we'll discuss next week. So again, no long-range order in membranes, 2 degrees of movement, of freedom of movement. Now, in the last 10 or 15 years, a slight change in the model has come about with the discovery that certain lipids, specifically cholesterol and sphingolipids, form a separate phase in the membrane. So it's a kind of a patch. These lipids organize in a patch, and these patches, like little icebergs, even when the membrane is otherwise fluid, float around independently within the plane of the membrane. And some proteins, but not all, have an affinity for these so-called raft structures. And they then float around along, embedded in this raft, and are not generally, for certain proteins, free to move into the phospholipid portion of a bilayer. So this is not an extrinsic factor like the spectrin network. It is rather an intrinsic factor. So that, in other words, there is some order within the bilayer, although if one looks at these raft structures in a living cell, they tend to be fairly randomly distributed, like little islands floating around independently within the plane of the bilayer. Though being very much larger, their rate of diffusion is slower than that of a phospholipid molecule, or even of a protein molecule that is otherwise embedded in the bilayer. So these are generally called glycolipid rafts, because the sphingolipids contained within them are glycosylated sphingolipids. Okay, any questions on this before we move on to transport of small species, small charged species? Yes? It's just an affinity that cholesterol has for certain glycolipids. Not well understood. There may be a charge that attracts the hydroxyl group of the cholesterol to the glycans on sphingolipids, but they form a separate liquid phase within the plane of the membrane, even in membranes that are otherwise completely fluid. And these rafts likely have special functional qualities. It's been suggested that they enrich signaling protein complexes, receptors, and you can then use the affinity of 2 different proteins for a raft to cluster 2 different proteins in close proximity, whereas were they floating around in the phospholipid bilayer, they might be less able to come into contact by diffusional collision. Yes? They differ quite dramatically. They're quite dynamic. They merge with one another. They separate by fission, and they're of course under some metabolic control. You can dissipate them by removing the cholesterol from membranes, but they're really quite dynamic. So things are believed to pop in and out of the raft. Okay, good. So now let's go on to begin a discussion of transport. And we'll see it in two different contexts. The first context is we'll simply discuss the broad categories of transport. And then later on, when we talk about electrically active membranes, we'll talk about rather specific and, of course, terribly important channels that mediate the generation of an action potential and the movement of an action potential down an axon of a nerve cell, and then the transfer of information from one nerve cell to another or between nerve and muscle cells, again relying on channel proteins, either voltage-gated channel proteins or ligand-gated channel proteins. But that'll be in a few weeks. In the first instance, one can consider channels or permeases in two broad categories. The simplest channel is one that mediates passive transport. We've already talked about one. It's the porin protein in the outer membrane of the mitochondrion or in the outer membrane of E. coli. It's a stable pore that limits permeation to molecules below several hundred Daltons in size. It's non-selective, tends to prefer polar species. It's just a conduit through which small molecules can pass, and the concentration on both sides of the membrane will achieve equilibrium. There's no concentrative transport mediated by this kind of passive transport through pores. Now, another somewhat more interesting form of transport that's passive is mediated by something called a facilitated carrier. A facilitated carrier is a protein that recognizes a particular small molecule, glucose, for instance. There are facilitated carriers that mediate glucose transport into our cells. And by itself, without any intervention of some source of energy, the glucose facilitated carrier will permit glucose to be equilibrated from outside to in. So if you have a meal and you're digesting your starch into glucose, it's taken up initially by cells through these glucose channels, and glucose concentration is equilibrated. But as we'll see later on, possibly my next lecture, that same carrier that doesn't use energy directly can be driven by an external source of energy in the form of a different gradient to concentrate glucose against its concentration gradient. But the simplest thing to remember about a facilitated carrier is that it recognizes a particular molecule or charged species. It allows it to be transported selectively. Nothing else will go through that channel, and it will not, by itself, concentrate. It will merely equilibrate. So no energy implied. All right, now, we're going to spend a lot of time talking about active transport. And active transport means the concentration of a charged or small species, small polar species, against its concentration gradient. The incorporation of an amino acid into a cell or a sugar molecule into a cell against a very steep chemical concentration gradient. This requires energy. The energy can be imparted either directly, and we'll talk about the major categories of that in a moment, energy-driven direct, where ATP acts directly on the channel protein itself, or indirectly, where the active transport is mediated by the connection of, for instance, a facilitated carrier to a gradient of something else that it can sense and exploit. So two forms, direct or indirect or linked. And I'll explain those as we go on. All right. Well, let's spend some time talking about the ATP-driven direct transporters. The majority of ATP-driven transporters can be categorized broadly into three types. The F type is the ATPase that I already told you about. It's the one in the mitochondrial inner membrane that has two parts, an F1 part that is a rotary engine consisting of three different ATP binding subunits that can move in one direction and hydrolyze ATP or move in the other direction and make ATP. And that F1 complex is linked to an integral membrane complex that contains a proton pore, and protons flow through that pore depending on whether ATP is being consumed, hydrolyzed, or synthesized. So it's a rotary engine linked to a proton gradient. The F-type ATPases. The mitochondrion represents one major species of F-type ATPase, but another one that we're going to consider in another couple of weeks is called the V-type, a similar kind of ATPase, but different organelle, also consisting of V1 and V0 complexes. The V-type ATPase is in vacuole membranes, and it's responsible for pumping protons from the cytoplasm into the interior of an organelle or a vesicle to make the interior of that organelle acidic. So this ATPase is not designed to synthesize ATP. It's designed to exploit ATP to generate an acidic interior in a compartment like an organelle called an endosome or the organelle called the lysosome, both of which we'll discuss in some detail in a couple of weeks. So these are the two major ATPases, and they're huge complexes, and the F1 complex has been solved at an atomic resolution, so the structure revealed exactly how it can move in two different directions to permit protons to move one way or the other. We'll spend a few minutes in a moment discussing the next type, the P-type ATPase. The P-type ATPase is a different kind of ATP-driven pump. P-type ATPases, unlike the F-type, involve the transfer of a phosphate from ATP, the third or gamma phosphate from ATP, onto the carboxyl group of an aspartyl amino acid in the active site of the enzyme. So ATP is hydrolyzed, but the phosphate doesn't just float away. It actually becomes covalently attached to form an aspartyl phosphate bond. We'll talk about that in a moment. And that activated state of the protein then permits the protein to change its shape to permit ions to move in one direction. So all P-type ATPases involve an aspartyl phosphate intermediate. One that we'll spend a lot of time talking about in the next few minutes is involved in transporting sodium and potassium in opposing directions in all animal cells, or virtually all animal cells. That's really one of the most important ATPases. Another type that we'll discuss very briefly in a few weeks is responsible for pumping calcium into muscle cells, into an organelle that's called the sarcoplasmic reticulum. And you'll see in muscle cells there's an internal organelle similar to the endoplasmic reticulum, and this organelle has a fairly high concentration of calcium in its interior space in the lumen, and that's because this P-type calcium ATPase pumps calcium from the cytoplasm into the interior of this organelle to store it, where it's used as a reservoir to signal muscle contraction. We'll talk about that later on. Again, that enzyme, for which there is a nice atomic resolution crystal structure, employs an aspartyl phosphate intermediate. So it's a different category, and they can all be recognized in new genomes that are sequenced because they have certain sequences, amino acid sequences, that are in common, that distinguish P-type and F-type ATPases. A third type that we will have only a moment to discuss, very interesting type, is called the ABC type, or ATP binding cassette, ABC. These were recognized many years ago by people who were studying why tumors become resistant to chemotherapeutic agents. So when you are a cancer victim, you take usually one or another of a toxic drug which comes into cells, is taken up into cells, and often will inhibit, for instance, DNA replication in those cells, selectively killing cells that are rapidly growing and dividing. So they're effective agents, but unfortunately, with time, cells emerge from within a tumor that resist the action of these drugs, and the reason that they resist the action of these drugs is they elevate the level of expression of one of these ABC-like transporters that has the sinister effect of pumping the toxic compound back out of the cell. So tumors very often resist chemotherapy after a few months because these guys get ramped up by the cancerous cells to expel this toxic compound. It makes a lot of sense that cells would have permeases like this to get rid of noxious compounds. The normal low level of these ABC permeases will suffice under most circumstances, but if you're being treated with massive doses of some toxin to kill cancerous cells, then the cells naturally respond by ramping these things up. And that greatly diminishes the effectiveness of these agents. And so an oncologist will try to attack these permeases by adding some other drug that tends to plug up the channel in the ABC carrier. So it's a kind of chemical warfare between the tumor cells and the oncologist. Anyway, so these proteins are really interesting, and they crop up in many different circumstances. This is the one that led to the discovery of this type of permease. Again, it has an ATP binding site, so that implies that ATP directly drives the action of this protein. Let's turn for a more detailed discussion to the first P-type ATPase that was discovered in dog kidney. It's the enzyme that's responsible for pumping sodium out of cells and potassium into cells. So most of the pathways in the cell prefer, as a cation, potassium. They don't like sodium. They tend to be less active in the presence of sodium. So cells expel sodium from their interior, maintaining a fairly high extracellular concentration of sodium, and they import potassium from the environment, maintaining a nearly equally high concentration of potassium inside the cell. And this exchange, which as you'll see in a moment is an unequal exchange of ions, is generated by a P-type ATPase called the sodium-potassium ATPase. If you turn to the diagram page from today, in the middle of the first diagram page, the only diagram page, you'll see a kind of a crude characterization of the structure of the sodium-potassium ATPase. Let me reproduce it on the board for you. So this is obviously an integral membrane protein. It was really very difficult to isolate, but a Danish biochemist succeeded something like 50 years ago to finally isolate this protein in pure form. And it consists of 4 subunits. A heterotetramer with 2 copies of an alpha subunit and 2 copies of a beta subunit, so alpha 2, beta 2. The alpha subunit has all of the business part of the molecule that we'll discuss. It's a protein that spans the bilayer with portions that face the outside of the cell. It's a glycoprotein. The beta subunit is also an integral protein, but it only is embedded into the outer portion of the plasma membrane. So this is beta, and this is alpha. All of the relevant business parts of this enzyme are located in the alpha subunits. Specifically, the ATP binding sites are located on the cytoplasmic face of the plasma membrane. and the sodium binding sites, the sites that will be occupied as sodium begins to be expelled from cells, are also on the inner surface of the cell, of the protein, whereas potassium, at least initially, 2 potassium sites are found on a portion of the alpha subunit that is exposed to the outside surface of the cell. So now here's what happens in the cycle of events mediated by this protein. In its initial resting state, ATP is bound to one or the other of the 2 subunits of the alpha-2 complex. Sodium ions, and I should say that this ATP is always complexed itself to magnesium. 3 sodium ions bind to as yet ill-defined sites somewhere on this surface close by the ATP binding sites. And then an aspartate residue in one or the other of the alpha subunits near the ATPase active site acts upon the 3rd or gamma phosphate, transferring it to a carboxyl group, the aspartate carboxyl group, to make the aspartyl phosphate intermediate that characterizes the action of P-type ATPases. That act, the hydrolysis of ATP and the transfer of the gamma phosphate to this slightly lower but still high energy phosphate ester linkage, changes the shape of the channel to permit the sodium ions to be expelled. So you can imagine it as a kind of a rocking action where sodium ions, if you look at the bottom of my hands, sodium ions are bound to a cleft facing the cytoplasm in the resting state, but when ATP is hydrolyzed, the enzyme changes its conformation to expose, in some way, the sodium ions to the outside of the cell, and they can then diffuse away. So that first part of the cycle permits 3 sodium ions to be expelled. At the same time, in that intermediate state, potassium ions bind to some other site now exposed on the cell surface, and that the binding of potassium ions stimulates the hydrolysis of this phosphate on the other side of the membrane. Phosphate then is returned as inorganic phosphate back into the cytoplasm where it can be scavenged to make more nucleotide. And that act, the hydrolysis or resolution of the aspartyl phosphate bond, restores the enzyme to its resting state with the concomitant exposure of those potassiums now to the inside of the cell. So again, the enzyme rocks back and forth. It's a 2-state engine. In the first state, the activated state, sodiums are expelled. When it's returned to the resting state, potassiums are imported. And then, of course, the cycle continues over and over again, many thousands of times a second. Over time, an electrically active cell can build up hundreds of millimolar concentrations of sodium on one side of the cell and a nearly equivalent concentration of potassium on the other side of the cell. Now, one special feature, very important, really critical feature of this channel is the unequal number of cations that are pumped for each cycle. I emphasize that 3 sodiums are expelled for every 2 potassiums that are taken up. That imparts, along the inner surface of the plasma membrane of electrically active cells, a net negative charge. There is locally an unequal distribution of cations. Over a long range, the concentrations are essentially equivalent, but over the range of no more than, let's say, 10 angstroms along the inner surface of the plasma membrane, there is a unequal distribution of charges, a net negative charge imparted to the inner surface of the plasma membrane, which makes these membranes electrically active. That is a gradient that we'll see is exploited quite liberally for the transport of lots of other molecules and is used to generate an action potential in the transmission of nerve impulses along an axon. Okay, so then the net unequal flow of these cations creates this charge. Thus, the sodium-potassium ATPase is said to be an electrogenic pump. Very important property. It's kind of it's there to kind of charge a battery. It's making an electrical charge that will keep an electrically active cell, impart a charge that can be used over a period of many minutes or hours, drawn down to transport other molecules, as we'll see in just a bit. Okay, so this is a really critical enzyme. Unfortunately, this particular P-type ATPase has resisted the ability of crystallographers to achieve an atomic resolution structure. The calcium ATPase has been solved, but the sodium-potassium not. So any of you who are looking for a Nobel Prize, all you have to do is that. Good luck. So now let's talk a little bit experimentally about how you study this kind of an enzyme. You can purify it, you can solubilize it in a detergent, and you can put it back into a vesicle. But now how do you study the activity of this enzyme and its orientation in a membrane, either in a native membrane or in a reconstituted membrane? Well, there are two useful reagents that I can mention that have been applied to study the protein that act at different surfaces. The first is an oxidized form of vanadium, which, for reasons that can't readily be explained, works nicely on certain P-ATPases along with ADP to coordinate and bind at an ATP binding site. So the ATP binding site on the cytoplasmic face of the alpha subunit of the sodium-potassium ATPase will bind in a coordinated manner vanadate and ADP, and obviously there's no beta-gamma phosphoanhydride bond, so there's nothing to hydrolyze, but the binding of the ATP site by these two molecules is sufficient to lock the enzyme in an inactive, dormant, resting state. It's a potent inhibitor. So, useful because this vanadate doesn't tend to interfere with too many other ATPases. It's, for reasons, again, I can't explain, fairly selective for this particular ATP binding pocket. All right, so suppose I told you that I had a cell that I knew had the sodium-potassium ATPase, and I wanted to study the action of this potential pharmacologic agent, and I added a few micromolar vanadate and ADP on the outside of the cell. Would it or would it not inhibit the enzyme? I heard a no. Who's brave enough to provide an answer to that no? Was that you? Okay, why not? It's on the wrong side of the membrane. What do you mean? Why doesn't it just swim across? You can't diffuse. Vanadate, cells don't want vanadate. And you have to have a very high concentration for vanadate to be fooled into being taken through a phosphate channel. So this is another setup, another kind of reagent, in this case an inhibitory reagent, that can be used to diagnose the accessibility, the exposure of this particular ATP binding site in an even, crude membrane. Okay. Well, that works on one side, but now there's an even more sinister compound, a type of compound that we'll talk about a little bit next time too, called wabane. It's a a venom toxin. It's a member of a larger family of compounds called cardiac glycosides. And you'll see in a moment, or actually next time, why they're referred to as cardiac glycosides. They tend to be potent inhibitors of cardiac muscle contraction. So they are extremely toxic and toxic neurotoxins. Wabane and other cardiac glycosides bind not on the ATP binding site, surface of the membrane, but they bind somewhere in the vicinity of the potassium binding sites on the external surface of the cell. That's why they're so toxic. That's why nature has evolved these nasty toxins and venom. They bind to this site, and curiously, what Wabane does, quite unlike what Vanadate does, is it permits the first step in this cycle to be executed, namely the formation of the aspartyl phosphate bond. But it prevents, when bound to this site, it prevents the hydrolysis of that aspartyl phosphate bond. Thus, the enzyme can't return to its resting state. So in the presence of Wabane, the first 3 sodium ions are expelled, so the enzyme rocks to look like this, but then either potassium can't bind, or once bound, it can't return to its resting state because the aspartyl phosphate persists. And so the next part of the cycle is blocked. 2 potassiums can't enter the cell, and so the whole process comes to a halt. You may have expelled the first 3 sodium ions, but the next cycle and the next cycle and the next cycle are inhibited. So the net flow of cations by this channel is effectively blocked, essentially as effectively as Vanadate, but it works on the other side of the membrane, the extracellular surface. Okay. Any questions on this really important enzyme? Okay. So let's continue. Just a few more minutes. Now, I want to tell you about how the existence of this sodium gradient provides a rich source of energy, in a way, linked energy, to permit a cell to take up or to expel other charged species. And we'll return, just for the moment, to the glucose channels that I told you about half an hour or so ago. So we all have, in fat cells and muscle cells and liver cells, virtually everywhere, we have a series of different kinds of glucose channels. And these are facilitated carriers. So glucose can come in or go out and will equilibrate through this channel very stereoselectively, unless there happens to be in the cell a high external concentration of sodium. So if the external concentration is much higher than the internal concentration, then of course sodium will want to try to flow down its concentration gradient if there's a way to sneak into the cell, and nature has provided just such a path as a pore through the facilitated glucose carrier. Glucose and sodium are symported through this channel together in a stoichiometry of 1:1. So the existence of this gradient now allows the glucose channel to become, in a way, an active, but in this case, it's linked to a gradient, so it's a linked form of active transport. And as long as the sodium gradient is maintained, glucose can now be taken up to a very much higher concentration inside the cell than outside the cell. Because the magnitude of the sodium gradient, being hundreds of millimolar on the outside of the cell, is much greater than the magnitude of the glucose gradient. So as long as this gradient is much steeper than this gradient, and the chemical concentration of sodium correspondingly much higher than of glucose, then glucose can be taken up to, from 1 millimolar outside of the cell to 20 millimolar inside of the cell, driven by this hundreds of millimolar concentration of sodium. So this is an example, a very powerful, important example, one of many called a symport carrier, and it relies, as I said about 20 minutes ago, on a facilitated carrier that is turned into an active transporter. Okay, on Friday we'll continue with more examples of this and how this seemingly noxious compound, wabane, can actually be used to treat patients. So, more later.